Dear students, myself B. Suresh Kumar, lecturer in zoology, working in SRR Government Arts and Science College, Karimnagar of Telangana State, affiliated with Shatavahana University. Yesterday, I created a channel that is Zoology for All. It is very made easy to students for degree and PG students also. So the channel name is Zoology for All. Remember and subscribe it. So today we are going to learn online classes during COVID-19 unlock period. It is day 2 and second lesson. Yesterday we discussed one content in zoology, that is digestion content. So today we are going to learn second lesson of SEM 5 students. It also useful to SEM 3 students. The SEM 3 students going to appear revised syllabus in the SEM 3 also the same content present. So the lesson useful to both the SEM 5 and as well as SEM 3 students. So now we are let us going to recapitulate the yesterday topic. So first of all, we learned about definition of nutrition in yesterday class. So later the intake food, whatever food we consume, the organism consume, the animals consume, so that food contain various kinds of chemical substances, they called nutrients. So definition of nutrients we learn, and as well as significance of nutrients. What is the use of nutrients? Thus, provides energy, body builder, regulator, like that. We learned the significance of nutrients, and as well as the organic and inorganic constituents presence of nutrients. Yesterday we discussed organic contents that are present under nutrients are carbohydrates, proteins, lipids, nucleic acids, vitamins. All these are comes under category of organic constituents, and minerals, water comes under inorganic constituent. In the same way, as per the consumption and involvement, the nutrients are categorized macro and macronutrients. The macronutrients are carbohydrates, proteins, lipids, nucleic acids, and micronutrients are minerals and vitamins. And after that we discussed types of nutrients based on function. Based on function, the nutrients are categorized as energy producers and body builders, body regulators, like that. The nutrients are classified. And after that we completed definition of digestion, the breakdown of complex food molecule into simple absorptive material, simple molecules, called what? Digestion. So in digestion, larger size molecules are undergoes hydrolyzed and they break down into simple absorptive material. So that is digestion. And modes of digestion also we completed. According to location, we finished the modes of digestion. So as per location, if for example the nutrients digested outside the cell, that is called extracellular digestion. If they digested inside the cell, that is called intracellular digestion. We are going to discuss in detail today about intra and extracellular digestion. In previous lesson, peripherally we completed intracellular digestion. So the intracellular digestion is a primitive phenomena. It is a primitive digestion. Most of the primitive animals exhibit intracellular digestion. If we observe animals, various grade animals are present in animal kingdom. They exhibit cell grade and tissue grade, organ grade and organ system grade, like that. Different level of organisms are there, animals are there. According to their grade, development is present. So the primitive animals are cell grade animals, and such cell grade animals exhibit intracellular digestion. And as well as the cell grade animals, what are comes under cell grade animals? Porifera individuals. First protozoans are comes under this category, and porifera individuals are comes under this category. So all these are exhibit intracellular addition. You observe that visual, right side the visual is present. The food content, the food material, the food particle enter across the cell membrane and into the cytosol and break down as a simple absorptive material inside the cell, that is represents intracellular digestion. So the intracellular digestion mainly include intake of the food material into cell, that is mediated by two ways. One is phagocytosis and pinocytosis. You observe phagocytosis. So you observe this visual. The solid content food particles are entered across the cell membrane, and by the invagination of cell membrane along with the food particle, it gave rise a vacuole that contain food particle. So that vacuoles are called phagosomes. The entire phenomena is termed as phagocytosis. Let us discuss one more event related to intracellular addition, that is pinocytosis. You observe pinocytosis phenomena. It is very simple phenomena. In pinocytosis phenomena, the food contents are liquid material. The liquid particle, the liquid material trapped into the cytosol across the cell membrane. So in this phenomena also, the cell membrane invaginated and the liquid contents are enter into the invaginated part of the cell membrane, and prominent vacuole is produced that contain liquid content. So this vacuole, you observe that vacuole, blue color liquefied material is present. That vacuole is called pinosome. So by phagocytosis, phagosomes produced in cytosol. By pinocytosis, pinosomes are produced. So both are the phagocytosis, pinocytosis results the intracellular digestion. It is a primitive digestion. So let us we observe the intracellular digestion takes place in which individual. Already yesterday we discussed a lot, but even though once again in detail today we are going to discuss in this lesson. So the first example, you observe digestion in amoeba. Amoeba is a protozoan animal, unicellular animals, cellular grade individual. It is cell grade individual. So you observe that amoeba food particle, it shows holozoic nutrition. What is holozoic nutrition? Consumption of the solid form food material is called holozoic nutrition. So the amoeba consumes the solidiform food particles with the help of pseudopodia. So it captures the food particle through pseudopodia The food particle is solid form, it is a holozoic, it comes under holozoic nutrition. Whenever food particle contact this cell membrane, immediately the pseudopodia evolved around the food particle and captures the food particle into cytosol, by which prominent vacuole is produced. You observe third visual, the food particle along the food particle invaginated with cytosol, and it's surrounded by lysosomes. And next, fourth visual, you observe all the lysosomes are surrounded the food vacuole and releases acid hydrolytic enzymes. Already we discussed that the lysosomes contain more than 40 variants of acid hydrolytic enzymes. So such acid hydrolytic enzymes break down the food particle into minute molecules, by which the entire intracellular addition completed. The digested food material to be absorbed into cytosol, the remaining undigested food material to be ejected outside. You observe last visual, sixth visual present in this PPT. You observe the clearly present the residual bodies, undigested particle ejected by the body surface. So that is addition. So next, let us we will observe one more example to intracellular digestion. So that is digestion in paramecium. So paramecium is commonly called slipperan molecule. It is a ciliary filter feeder. With the help of cilia, it traps the food particles, enter into oral groove. Clearly pink color obliquely seated oral groove is present in visual. You observe the food particle lead into cytostome, cytostome to cytopharynx, and ultimately food vacuoles are separated. That food vacuoles, phagosomes, are undergoes intracellular digestion in the body of the paramecium. You observe this visual, the intracellular digestion paramecium express. So it is a ciliary filter feeder. You observe cilia movement in the visual. And next, you observe this visual, the food particles are present in the surrounding, trapped into the cytosol, and the oval shape vacuoles are phagosomes. They are undergoing the digestion process. So this is a clear visual that represents the intracellular digestion paramecium. Next, we are going to discuss one more example to intracellular digestion, that is sponges, belongs to phylum Porifera. So they also exhibit cellular grade. They are multicellular animals, but actually their body constructed with more than single cell. Even though they won't consider under tissue grade, because the cells never aggregate in them, they arranged as isolated fashion in their body. That's why they are called cell grade individuals. Of course, they are multicellular animals, but tissue grade is absent. So in these individuals also, same intracellular addition to be expressed. A better example provided in visual, that you observe sponge, that is central spongiocele is present. Through ostia canal system, through water current, the food particles are enter into the spongiocele. Already all these things we learned in invertebrate phyla. So all these food particles captured, trapped by the flagellated cells. The flagellated cells are called choanocyte. You observe the visual, violet color cell is clearly present. The pink color fiber-like extension is present. So that is flagella. These flagellated cells are called collar cells, otherwise choanocytes. These choanocytes intake the food material. Yellow color food particles are present. You observe the yellow color food particles are enter into the cytosol of the collar cell choanocyte and ultimately undergoes intracellular digestion. So it represents one more example. And next, besides it, not only in animals, in higher animals also some vital activities are achieved by intracellular digestion. For example, uh, phagocytosis by the WBC. So that is also uh, one kind of better example to intracellular digestion. So the intracellular digestion mechanism in detail already we discussed, but you observe this representation diagram clearly. So that is eukaryotic cell produce phagosome associated with the lysosome going to release acid hydrolase enzymes. That acid hydrolase enzyme, after completion the action of acid hydrolase enzyme, the contents are absorbing into the cytosol. It is a better example, better visual to intracellular digestion mechanism. So next, now this is you observe this animated diagram. The first one represents eukaryotic cell, so that is food particle. So next, second visual represents the lysosomes. So they are under food vacuole. How, in what pattern they are going to interacting with food particles. So this visual represents. So this is mechanism of intracellular digestion. And next, we are going to discuss about extracellular addition. So one mode is intracellular addition and second mode is extracellular addition. So intracellular addition is a primitive method. The primitive individuals are exhibit. Extracellular addition, it is an advanced mechanism. So in this phenomena, the food particles to be digested outside the cell. The food particle never enter into the cell. After completion digestion, they can enter into the cell, but before digestion, never enter. But intracellular digestion, not like that. Before completion digestion, they enter into the cell, but in extracellular digestion, they never enter into the cytoplasm before completion digestion. So you observe this visual clearly. The blue color particle represents food particle. The yellow color rectangular shaped cell, that represents rectangular shape body represents cell. So the secretory organelles are secreting some contents outside the cell. They are nothing but enzymes. That enzymes are reacts with the food particle and digest that food particle outside the cell. So in that way, entire food particle completely digested outside the cell. That is extracellular digestion phenomena. Is it clear? So what difference you find out between intra and extracellular addition? In intracellular addition, the hydrolysis process extended inside the cytosol. But in extracellular addition, the hydrolysis process extended outside the cell. Of course, the addition is a hydrolytic process. Both are hydrolytic processes, but the location is different. Outside one and inside one. So intracellular addition, that is, and extracellular. Is it clear? So next, the extracellular addition, it is advanced mechanism. So the advanced individuals are exhibited. So cell grade individuals are upgraded as tissue grade individuals during course of evolution. You observe the animal kingdom, the first phylum is Porifera. In Porifera, sponges are there. they are cell grade individuals. In core of evolution, these cell grade individuals are upgraded as tissue grade individuals. So the porifera individuals are evolved as Cnidarians. That is next phyla. So Cnidarians are what? Tissue grade individuals. So the tissue grade individual onward up to organ and organ system grade individuals, all individuals are exhibit what? Extracellular addition. So advanced group of individuals exhibit this extracellular addition. So first after completion cell grade individual, first grade individuals are tissue grade individuals. So the tissue grade individual, whatever comes under tissue grade individual, Cnidarians or comes under tissue grade individual. So next in tissue grade individual, if we observe, they performs both dual mode of digestion, intracellular and extracellular addition. So this is a significant character of Cnidarians. Remember, asked in entrance examinations also. In case of protozoans and porifera individuals, exclusively intracellular addition is present. But in tissue grade individuals, Cnidarians, they are showing both intra and as well as extracellular addition. Are you know about the examples of Cnidaria? For example, Hydra, Aurelia, commonly called as jellyfish, Rhizostoma, like that, corals. So many examples we learned in invertebrate phyla. So let us in this visual one example provided, that is Hydra. The fiber-like, filament-like structures are tentacles. In between tentacles, prominent mouth is present. Through the mouth, the red color arrow mark indicates the ingestion. So the food particles, they also exhibit holozoic nutrition. The food particles are enter into the hollow cavity of the body. The central hollow cavity is called Gastrovascular Cavity. So you observe the body wall of the Cnidarian, the cylindrical shaped cells are present. The body wall constructed with the cylindrical shaped cells. They are diploblastic individuals. Their body wall constructed with two layers, epidermis, outer layer, and gastrodermis, inner layer. So two layers are present. During embryonic condition, during embryonic development also they exhibit two germinal layers, ectoderm and endoderm. Hence they comes under category of diploblastic category. Hence we classified them in diploblastica grade. Okay, so you observe the gastrovascular cavity. In the gastrovascular cavity, whenever food particles are entering gastrovascular cavity, you observe this visual. Cyclops is consumed in gastrovascular cavity of the Hydra. After that consumption, like sponges in the wall of the Cnidarians also, some flagellated cylindrical cells are present. They are called nutritive epitheliomuscular cells. In case of sponges, they are called collar cells, choanocytes. But in case of Cnidaria sponges, the flagellated cells are nutritive epitheliomuscular cells. You remember, asked in entrance examinations. So, in the point of entrance examination, the technical terms and the significant cells and significant features of the animals are most important. Based on it, definitely the questions to be raised in entrance examination. So here I am discussing very clearly. So with visuals, you have to listen my words and as well as follow the visual clearly. You can understand the topic, and afterward if you read any textbook related to zoology, then immediately you uh, able to describe the answer in the examination, and you able to find out the correct answer in multiple choice questions also. Is it clear? So, that gastrovascular cavity you observed, cyclops conjunct you observe, and the inner layer cells are nutritive musculoepithelial cells. And you observe this visual, it is section diagram. Clearly, flagellated cells are there. They called nutritive epithelial muscular cells. In front of them, the food particles are present. The food particles are going to intake in that cell and going to digest inside the cell. So that is intracellular addition. In the same manner, in between the nutritive musculoepithelial cells, enzyme secreting cells also present. They secrete directly enzymes into the gastrovascular cavity. So, part of the food to be digested inside the cells, that is intracellular addition. Part of the food to be digested in gastrovascular cavity. So that comes under extracellular addition. So hence the Cnidarians are showing dual mode of digestion, intracellular addition and as well as extracellular addition. Very clear? Yes. Next you observe one more important thing. They not only exhibit a dual mode of digestion, so you observe the terminal opening of the hydra contain only single opening, that is mouth. Through the mouth the food particles are trapped into the gastrovascular cavity. So it mediates the ingestion, and undigested food particles also from the gastrovascular cavity they ejected outside. Ejection also mediated by the mouth. So mouth also useful for dual purpose, for ingestion purpose and ejection purpose. So digestion is dual purpose and mouth also used for dual purpose. So that is the significance of the Cnidarians. So let us we observe the upgraded animals after Cnidaria. After Cnidaria, the subsequent after Cnidaria, the subsequent phylum is Platyhelminthes. So they shows what organ and organ system grade. First time the digestive system developed in Platyhelminthes animal in the core of evolution. The evolution is a sequential process. Simple organisms are turned into complex organisms. So in this process, in this gradual process, there is no digestive tract in the Cnidarians. But if we observe the Platyhelminthes individuals, prominent alimentary canal, digestive glands are developed in the Platyhelminthes individuals. So first time digestive system developed in Platyhelminthes individual. So from Platyhelminthes onward, subsequent phylum individuals, the digestive system greatly developed according to their feeding habitats and according to their requirement. It attained level. You observe this visual, the alimentary canal in Helminthes already we learned in invertebrate phyla, that is the planaria, dugitia, flatworm. The mouth is present at mid part of the body. You observe head is present, one pair eyes are present, one on either side. But you observe mouth outside the body at terminal end of the proboscis. The mouth is present. The mouth leads into pharynx and it leads into alimentary canal. That alimentary canal, branched alimentary canal is present, that extended all over the body. But you remember here in Helminthes also only mouth is present, but anus is absent. So they expect a single opening like Cnidarians, but comparatively with the Cnidarians, they are developed individuals. they attained a prominent digestive system. So after helminthes, next nematodes are there. That nematodes exhibit tube within tube body organization, prominent alimentary canal, and they possess mouth and anal opening. First time anal opening started in nematodes, and after that nematoda annelids are there. The phylum Annelida, a major example, earthworm, you observe earthworm contain prominent alimentary canal. You observe that visual yellow color track is present, tube is present. It is also tube within tube body organization. Prominent mouth is present. Prominent digestive tract is present, associated with the digestive glands. So, the phylum to phylum, how much development going on in the digestive system, you have to verify. After annelids, arthropods, you observe insects. Better example to it, insect, arthropods. They also contain prominent alimentary canal. Right, next we are going to discuss the digestive system in vertebrate animals. So the animals are categorized mainly two groups, invertebrates and vertebrates. In case of invertebrates, notochord is absent, vertebral column is absent. They grouped as invertebrate from porifera phylum onward up to echinodermata, up to hemichordata. They consider as what invertebrates. And next, after that, subsequent groups or group is chordates, in which prominent notochord is present. In chordates, those exhibit prominent vertebral column, they referred as vertebrates. Invertebrates, the first group of the vertebrate fishes. So you observe digestive system in fishes. Prominent alimentary canal seated, you observe. Next, after fishes, amphibians. Better example to amphibians, frog. It also contain prominent alimentary canal. And another group is reptile, also contain prominent digestive system. And next, another group, aves, contain prominent alimentary canal along with the digestive glands. And higher animal group is mammals. So in mammals also developed digestive system is present. So in this way, in core of evolution, the digestive system also attain various changes, simple to complex form. So you first observe platyhelminthes. First, in platyhelminthes, the digestive tract is very primitive with a single opening mouth and few separation is there. But after platyhelminthes, nematoda, two openings, mouth and anal opening is present. Some other organs also associated. Muscular layer also first time associated with alimentary canal in nematodes. So next, regional specialization also. Next, annelida. In annelida, well developed. Like that, you observe up to mammals how much development present in alimentary canal. You observe complexity attained. They attained more complexity, simple to complexity. So with this, we completed intra and extracellular digestion modes in all these individuals. And next lesson, we are going to discuss subsequent topic, that is digestive system of human being, organization of digestive system of human being. So I think I am explaining these topics from basic to complex. So this content utilized for all students, not only for degree PG students, and useful for all students. So So it is better useful to all. It is better useful to all competitive exams also. So from basic to complex, I am going to explain. So you have to follow the all these lessons sequentially from the beginning, and you have to subscribe the channel compulsory. And next, stay home and stay safe in the lockdown period. Right, thank you, all the best. Dear students, myself B. Suresh Kumar, lecturer in zoology, working in SRR Government Arts and Science College, Karimnagar of Telangana State, affiliated with Shatavahana University. Yesterday, we created a channel to learn zoology contents. SEM 5 students, that is Zoology for All by Suresh. So whatever lessons I am daily uploading in the channel, it is very useful to the graduate students of SEM 5 and as well as postgraduate students also. So don't miss the lessons sequentially. You have to watch the lessons in the channel, and definitely compulsory you have to subscribe. So next, in the lockdown period due to the COVID-19, we are conducting online classes. So in that online classes, the curriculum belongs to SEM 5, is physiology and biochemistry. In that, the first lesson of the unit 1 is digestion. In last class, we discussed about introduction about digestion. So these topics are covers the syllabus of SEM 5 and SEM 3 also. Why? Because our Shatavahana University revised the syllabus to semester 3 students. So in which also the same contents of SEM 5 compiled in SEM 3. So you have to, the SEM 3rd students also must follow these same contents. So today I am going to explain about in detail the process of intra and extracellular addition. So before going to learn about intra and extracellular addition process, first I am going to recapitulate the topics of the lesson 1. Yesterday we finished about definition of the nutrition. What is nutrition and what are nutrients? The chemical substances that are present in the food considered as nutrients. Already, what is nutrition? Collection of the food material by the animals are called nutrition. And next, significance of nutrients. What's the use of nutrients? They are useful for provide energy, for body growth, repair the tissue, like that. Significances we learned in lesson 1. And the nutrients, whatever present in our daily consuming food, that nutrients contained organic and as well as inorganic constant, isn't it? So already we discussed in last class The organic constituents are carbohydrates and lipids, proteins, nucleic acids, vitamins, and inorganic constituents are minerals and water. Based on consumption and based on involvement in function, based on their role, so these nutrients are again categorized two variants. They are macronutrients and micronutrients. Did you remember about macro and micronutrients? What are comes under macronutrients? Macro more. What are comes under macronutrients? Those nutrients we are consuming in large quantity and they plays key role. They plays a major role. So they called macronutrients. For example, carbohydrates, proteins, lipids, nucleic acids. They called macronutrients. What are micronutrients? We consumed them in little quantity and their deficiency results disorders also micronutrients. So what are comes under micronutrients? Vitamins and minerals. And based on based on function, the nutrients also categorize two kinds, three kinds. They are energy producers, example carbohydrates, bodybuilders, example proteins, and next energy reserves, lipids like that, and body regulators, example vitamins and minerals, isn't it? So are you clear? So all these things we completed in last class, and one more thing, that is definition of digestion. The digestion is a hydrolytic process. It is a physiological process, one of the physiological process. In this process, the complex food material break down into simple, minute molecules, absorptive molecule, liquefied molecule. The entire phenomena is called digestion. That is digestion definition. And according to mode of digestion, where they occur, according to location of digestion, the digestion categorizes two kinds. One is extracellular digestion and another one is intracellular digestion. So next, the what is intracellular digestion? Intra means inside. It is a primitive kind of digestion mode. The whatever food material consumed by the individual, the food particles, the food material completely enter into the cell of the individual, and the food particles are completely digested inside the cell under cytosol. So that is intracellular digestion. Let us we will discuss the what are the converse things, what are the converse aspects between intra and extracellular digestion. So based on differences, based on converse factors, mostly the questions to be raised in entrance examinations. Hence you have to follow that converse things between intra and extracellular digestion. Of course, in last class I explained clearly about intracellular digestion. Even though once again I am going to clear this topic with new visuals. So in this lesson, so the intracellular digestion is a primitive event found in expressed in primitive individuals like unicellular individuals, protozoans, and as well as multicellular animals, porifera individuals, sponges. The porifera group of individuals, they are multicellular animals, even though tissues are absent because the cells are never aggregated. If we observe the organization levels of the animals, The animals concerned to animal kingdom exhibit different levels of organization. First organization is acellular level, cellular level, and next tissue grade level, organ grade, and organ system grade level. Like the different grades are established during course of evolution. According to that grade, they exhibit this mode of digestion. So the primitive group of individuals are what? Unicellular individuals and multicellular individuals. These two individuals are exhibit the primitive mode of digestion, that is intracellular digestion. The intracellular digestion include mainly two processes. One is phagocytosis process, another one pinocytosis. Let us observe phagocytosis process. Clear visual is present in this slide. So outside the cell membrane, the solid form food material is there. The solid form food material captured through the cell membrane into the cytosol. So the food particles are dropped into the cytosol by the invagination of the cell membrane. Such phenomena is called phagocytosis. Mostly the unicellular individuals and multicellular individuals, and as well as some of the phagocytic cells of higher individuals also exhibit same phagocytosis phenomena that involve in intracellular digestion. That next another event of the intracellular digestion is pinocytosis. What is pinocytosis? In phagocytosis, the individual consumes the individual intake the solid form food material. But in case of pinocytosis, the individual consumes the liquid form of food material. You observe this visual. The blue color particles are present, orange color particles are present. The blue and orange color particles represents the liquefied food material. That liquefied food material captured, trapped by the cell membrane by the, you observe the invagination. The membrane going to invaginate into cytosol, and the food particles are completely trapped into the invagination. And later, the by the cutting the cell membrane, the oval shaped vacuoles are produced. So the vacuoles with liquefied food material is called pinosomes. So by the pinocytosis, pinosomes are produced. By phagocytosis, phagosomes are produced. By phagocytosis phenomena, the food vacuole, whatever present, that food vacuole with the solid form food material called phagosome. So by phagocytosis, the produced vacuole is phagosome. The pinocytosis results pinosome. So next, let us will discuss examples to intracellular digestion. One first example, you observe digestion in amoeba. Amoeba, unicellular individual, shows holozoic nutrition, consumes the solid form food material. In this visual, you observe prominent yellow color food particle is present. That is solid form food material. When it contact the plasmalemma, immediately around that food particle pseudopodia are formed. By with the help of pseudopodia, the food material captured into the cytosol. You observe this visual. The food particle going to capturing, and later the food vacuole phagosome is produced. That phagosome going to associate with lysosomes. You observe visual blue color particles are present. They are lysosomes, and they secretes various kinds of acid hydrolytic enzymes in food vacuole that serves digestion. And inside the food vacuole, the entire food material to be digested. After completion digestion, the digested material to be absorbed into cytosol. You observe fifth visual. And next, whatever food contents that never digested, they called residual bodies, undigested particles. That undigested particles, residual bodies contained vacuoles to be eliminated, ejected outside, that is called ejection. So you observe this phenomena in amoeba. The entire food material, entire food particles are completely uh, going to digest inside the cell. So that is intracellular digestion. And one more example, let us we will observe, that is digestion in paramecium. It is also unicellular individual, belongs to protozoan, commonly called as slipper molecule. In last class we discussed, you observe shape of the individual. It is a ciliary filter feeder. With the help of cilia, it trap the food particles towards ventral side. You observe oblique groove is present, that is oral groove. That oral groove leads into narrow canal, cytopharynx, cytostome and cytopharynx. Through that, the food particles are separated as food vacuoles. You observe this visual pink color particle represents the food particles. They are going to trapped into oral groove, cytopharynx, cytostome, cytopharynx, and then prominent food vacuoles to be produced. That food vacuole going to undergoes digestion in the cytoplasm. So intracellular addition. You observe this visual, it clearly explains the intracellular addition phenomena in paramecium. So this visual is very clear. You observe the movement of the cilia. With the help of cilia, it trap the food particle. Hence, they referred as ciliary filter feed. It's very important point. Definitely ask in entrance examination. Ciliary filter feeder means with the help of cilia, the feeding to be achieved, collection of food material to be achieved. So hence they called ciliary filter feeder. And next, you observe this visual real image of the paramecium. So in what pattern the food particles are going to intake through the oral route, and in what way the food vacuoles are rotating in the cytosol, and what fashion they are going to digestion, you have to clearly observe in this visual. So it is the intracellular digestion in paramecium. And next, one more example to intracellular digestion, one more group to intracellular digestion, sponges. In sponges also intracellular digestion to be occur. So if you observe the organization of sponges, they are primitive individuals. So they have a prominent canal system. Already in invertebrate phyla, you learned about the porifera individuals body organization. The body wall associated with the prominent canal system. In the canal system, the terminal aperture is ostia. Through ostia, the food particles are enter into canal system. So whenever water current flows along the canal system, the food particles are enter into the circulated into canals, and ultimately they enter into the hollow cavity that present in the body of the sponges. You observe this visual. In that visual, the central hollow cavity is called what? Spongiocele. So while during that time, the food particles are captured by flagellated cells. The spongiocele lined up with a specialized cells. They called collar cells, choanocytes. You observe this visual. Right side violet color cell is present. That is choanocyte, collar cell. Pink color filament is present. That is flagella. By movement of that flagella, they trap the food particles, yellow color particles, and the particles are ingested into the cell and ultimately digested inside the cell. So that represents the intracellular addition. So both the protozoans and sponges exhibit intracellular addition. Not only this group of individuals In higher animals also, some of the cells performs intracellular addition. For example, WBC activity you observe neutrophils. They exhibit phagocytosis, microscopic policemen. They engulf the bacteria, invaders, and performs the um, intracellular addition. You observe this visually, it clearly explains the mechanism of intracellular addition during phagocytosis phenomenon. So whenever food particle enter into the body, the lysosomes are releasing from Golgi complex and associating with the vacuole and releasing the enzymes going to digestion, intracellular digestion, absorption. You observe these two visual clearly represents the mechanism of intracellular digestion. Next, you observe this visual also. The first one represents the eukaryotic cell associating with food particle, that is ingestion phagocytosis phenomenon. The second visual represents the lysosomal activities, releasing enzymes and food particles going to digestion food vacuole. So these two visuals represents the intracellular digestions, clearly explains. And next we are going to discuss about subsequent content, that is extracellular digestion. Extra means outside the cell. So in this phenomena, the digestion completely occur outside the cell. You observe the converse things between intra and extracellular addition. In intracellular addition, the digestion occur inside the cell. In extracellular, outside cell. So the food particles without digestion enter into the cell in intracellular addition. But in extracellular addition, not like that. The food particle never enter into the cell without digestion. After completion digestion, they enter into the cell. But before completion digestion, never enter into the cell. That is the difference. That is the converse thing between intra and extracellular addition. Are you followed? Is it clear or not? In intracellular digestion, what happens? Intracellular digestion without digestion. Before the digestion, food particle enters into the cell. In case of extracellular addition, without digestion, never enter food particle into the cell. The food particle to be enter into the cell when after completion digestion enter into the cell. The digestion process occur outside the cell, that is extracellular addition. You observe this visual, it clearly represents the extracellular addition. The upgraded individuals are exhibit extracellular addition as advanced mechanism. The upgraded individuals after porifera are what? Cnidarians, tissue grade individuals, diploblastic individuals. Their body wall contain two layers. Their embryo exhibit two germinal layers. They are called diploblastic individuals. Upgraded individuals comparatively with porifera individuals, sponges, isn't it? So that upgraded group of Cnidarians exhibit extracellular addition. Not only extracellular addition, besides that extracellular addition, they also exhibit intracellular. They exhibit dual mode of addition, extra and intra. So after Cnidaria, after diploblastic animal, next upgrade group is triploblastic individual. In Cnidarian only tissue grade is present, but in triploblastic animal, organ grade, organ system grade is present, that starts from platyhelminths to up to chordata. According to upgrade, all these levels: cell grade, tissue grade, organ system, organ grade, organ system grade. So the tissue grade individuals exhibit both intra and as well as extracellular addition. Observe this visual. The blue color particle represents food particle. The yellow color box represents cell. From the cell, the lysosome going to releasing some particles outside, going to secrete some particles outside. They are nothing but what? Digestive enzymes. They break down, they hydrolyze the food particle outside the cell. So then after completion digestion, the particles to be enter into the cell. That is process of extracellular addition. So from tissue grade individual onward up to organ system grade individual exhibit the extracellular addition, but especially the Cnidarians or exhibit both intra and extracellular addition. You remember, it is entrance point of view, compulsory these things to be asked in entrance examination. It exhibit dual digestion, intra and extra. In what way it performs both intra and extracellular addition, let us will discuss. So are you do you have idea about examples to Cnidarians already learn in your classes? Hydra, Aurelia, corals, like that, so many animals are there. So Hydra is familiar example to Cnidarian. You observe that visual cylindrical shaped body, a terminal and prominent opening is present, that is mouth surrounded by filament like structures, tentacles. So by the movement of the tentacles, the food particles are captured. The red color line, red color arrow represents the ingestion phenomena. The food particles to be received from the mouth to a hollow cavity observed. Inside the body, a hollow cavity is present. The cavity is called gastrovascular cavity, surrounded by body wall. And the body wall, two layers are there. Outside violet color representation that represents epidermis, and inner yellow color layer represents the gastrodermis, endodermis. So they are diploblastic animals. So if we observe the gastrodermis, include cylindrical shape. You observe third visual, cylindrical shape flagellated cells are present. They call nutritive musculoepithelial. The organization is completely different in sponges and Cnidarians. In sponges, the spongiocele lined up by flagellated cells, they are collar cells. They are choanocytes. Remember, ask an entrance examination. But in case of Cnidarians, the gastrovascular cavity lined up with nutritive musculoepithelial cells. The cells name different, organization is different, shape, structure is completely different. Already we learned in anatomy of the Aurelia and Obelia also. So you observe second visual, through the mouth, whatever food particle ingested that will be enter into gastrovascular cavity. You observe cyclops animal is present in the gastrovascular cavity. So here these nutritive musculoepithelial cells, between nutritive musculoepithelial cells, glandular cells remain. They releases the enzymes and break downs the food particle and assist the intracellular extracellular addition, and as well as some particles are enter into the nutritive musculoepithelial cells and facilitate the intracellular addition. So in that way, in Cnidarians, both extra and intracellular addition takes place. Let us discuss after Cnidarians. The next group is Platyhelminthes. They are triploblastic animals. First triploblastic animals exhibit three germinal layers. So it is development phenomena in the core of evolution. These are also upgraded individuals. Organ grades and organ system grades started from Platyhelminthes. So here onward, the prominent digestive system developed. They also exhibit extracellular addition, but not intra. Complete, purely, exclusively extracellular addition exhibited from Platyhelminthes animals to chordate group. Exclusively intracellular addition exhibited by protozoans and porifera individuals. Dual mode, tissue grade individuals. And purely extracellular addition from Platyhelminthes onward, chordata group of individuals exhibit extracellular addition. So if we observe the digestive system development in these group of individuals, generally the animals are categorized two kinds: non-chordate group and chordate group. Non-chordate, also called invertebrates, no vertebral column present in them. Chordates, prominent Notochord is present. In some, that notochord replaces by vertebral column. They called vertebrates. So, if we observe non-chordate triploblastic and chordate triploblastic, the development of digestive system is completely different according to their feeding mechanism, according to their feeding habitats, according to their requirement. The digestive system attained complexity. First time the digestive system developed in Platyhelminthes. Will you exemplify the Platyhelminthes? Tinea solium, planaria, like that individuals. So you observe digestive system in invertebrate group of animals. First, let us will discuss in helminthes in what pattern the digestive system is present. This is Dugetia, planaria. Head is prominent, eyes also present, but mouth is present middle of the body. At terminal end of the proboscis you observe, and leads into pharynx, and the alimentary canal, branched alimentary canal is present. So in a tissue grade individual, mouth serves both ingestion and digestion there. Here also only single opening is present, alimentary canal container serves both ingestion and digestion. No anal opening is present. Digestive system is present but anal opening is absent. Remember, it is also important thing. Next, another group of invertebrate phyla, annelids, Nematoda, Ascaris. So the Ascaris individuals are upgraded than helminth individuals. They exhibit tube within tube body organization. Prominent mouth is present. First time anal opening is developed in Ascaris. They show tubular body. These are flatworms. Completely the body is compressed. You observe that visual. Next, after nematode annelids, they are advanced than nematodes. Shows tubular body, cylindrical shape body. Earthworm you observe prominent inside yellow color tube you observe, that is alimentary canal associated with glands also. So development, what pattern the development is occurring, going on, you observe. After annelid, arthropoda group is there. Verify one example, better example to arthropods, insects, prominent alimentary canal along with digestive glands. After arthropoda, mollusca also, more soft bodied individuals are there. Molluscans, example snails, unio, and mussels, all these are paralyzed, all these are comes under sepia, loligo, like that individuals are comes under molluscans. Of course, some individuals are externally surrounded by hard calcium carbonate made up of shell. Of course, that is a protection purpose, that is. But inside the body, you observe, they are soft-bodied animals. For their protection purpose, they enveloped by calcium carbonate shell. Mollusks means soft-bodied animals. In them also prominent gut and glands are present. After mollusca, echinodermata group is present, starfishes. In them also prominent alimentary canal, digestive glands are present. After echinodermata, the next invertebrate non-chordate group is hemichordata, baranoglasses. In them also alimentary canal is present. So in all non-chordate animals, from platyhelminthes to up to hemichordata, exhibit prominent alimentary canal and digestive glands, but the organization is different. In some organs are hugely developed, some organs are may not present. So according to their feeding habitat function, that is present. So next group of the animals in animal kingdom, chordates. They exhibit prominent notochord, postanal tail, pharyngeal gill slits, dorsal nerve cord. These are fundamental features of chordates. Already we learned in lower classes. So in them, the notochord are replaced by vertebral column. Some individuals, in some chordates, at the place of notochord, prominent vertebral column to be established. Such group of chordates called vertebrates. Of course, already we discussed all vertebrates are chordates, but all chordates are never vertebrates. For example, urochordates are there, cephalochordates. They never exhibit vertebral column. So all vertebrates are chordates, but all chordates are never vertebrates. So if we observe the chordate group, first group is protochordate. They also exhibit prominent digestive system. The next group is vertebrate. In vertebrate, the vertebrates are grouped as first level is fishes. You observe the fish organization, prominent alimentary canal associated with glands are present, and amphibians. Example to frog. Example to eat frog. You observe alimentary canal is present. The same manner, reptile subsequent group contain developed alimentary canal with glands, and subsequent group Aves contain prominent alimentary canal with digestive glands. Subsequent group is Mammalia. It is advanced group in vertebrate, so also contain digestive system along with digestive glands. Human being also comes under this category, mammals. So in that way, in this lesson, we discussed the development of digestive system in different individuals, and intra and extracellular addition phenomena discussed. So in next class we are going to discuss the organization of human digestive system. So next these topics continuously sequentially you have to follow. I think all these points, all these concepts I am going to explain in detailed manner from basics to complex, so that to be helpful to success in competitive examinations, and easily they will be that easily they understood. And after listening this lesson and observing these visuals, if you read any familiar zoology textbook according to your syllabus, then immediately you have to describe, you have to present a good manner in the examination, and that helpful to attain better success. And not only for a description, and easily you have to trace out the answer in multiple choice question. Why? Because the multiple choice questions completely analytical basis. Suppose from basic to complex manner, if you learn the content, then only you can trace out the correct answer. Otherwise you will misguide and attempt the wrong answer, that results the negative marks and other thing, and failureness to you. So you compulsory you have to follow the lessons sequentially. So we completed lesson 1 and lesson 2 in this unit, and next tomorrow we are going to discuss lesson 3. And daily sequentially you have to follow the channel and observe the lesson, and listen the lesson, and read the appropriate textbooks, whatever preferred, and so okay, that provides better success to you. Stay in home, stay safe.